The Physiology of Gastric Acid Secretion, a comprehensive, step-by-step exploration. Welcome, listeners, to today's in-depth exploration of human physiology, where we will unravel the fascinating process of gastric acid secretion. Our journey will illuminate the intricate dance between cells, signaling pathways, and regulatory mechanisms that together ensure the stomach functions optimally. Let us begin by examining the foundational cellular players in this process. Cellular mechanism of hydrochloric acid secretion by parietal cells. At the heart of gastric acid secretion are the parietal cells, also known as oxyntic cells, located within the gastric glands. These cells are equipped with a remarkable apparatus known as the proton pump, formerly the hydrogen-potassium adenosine triphosphatase. Through this pump, parietal cells actively exchange intracellular hydrogen ions for luminal potassium ions, thereby releasing hydrogen ions into the stomach's lumen. Simultaneously, chloride ions are secreted into the stomach through specific chloride channels, such as the cystic fibrosis transmembrane conductance regulator and the chloride channel 2. Once in the gastric lumen, hydrogen and chloride ions combine to form hydrochloric acid. A unique physiological side effect of this process is that for every hydrogen ion released, a bicarbonate ion is produced and transported into the bloodstream by the basolateral chloride bicarbonate exchanger. This results in a temporary rise in blood pH, known as the alkaline tide, which typically occurs after a meal. Regulation of gastric acid secretion. The secretion of gastric acid unfolds in three distinct phases: the cephalic, the gastric, and the intestinal phases. Each is orchestrated by specific triggers and mediators. Cephalic phase. Anticipation sets the stage. The cephalic phase accounts for approximately 30% of total secretion. This phase is initiated not by food entering the stomach, but by the anticipation of food, through sight, smell, taste, or even thought. The vagus nerve, also known as cranial nerve 10, is the star of this phase. It acts directly by stimulating parietal cells through acetylcholine acting on muscarinic type 3 receptors, and indirectly by triggering the release of gastrin-releasing peptide, which prompts G cells to secrete gastrin. Gastric phase. Arrival of food. As food arrives and distends the stomach, the gastric phase commences, responsible for around 60% of acid production. This phase involves a complex interplay of local reflexes, with more acetylcholine released to stimulate parietal cells, and the presence of peptides and amino acids stimulating G cells to release gastrin. Gastrin, in turn, encourages enterochromaffin-like cells to release histamine, which acts on histamine type 2 receptors on parietal cells, intensifying acid secretion. Intestinal phase. A balancing act. The final 10% of acid secretion is managed during the intestinal phase, which is initiated as partially digested food, or chyme, enters the duodenum. Interestingly, this phase is predominantly inhibitory. While small amounts of gastrin may be stimulated, hormones such as secretin, cholecystokinin, and somatostatin signal the stomach to decrease acid production, ensuring the digestive process proceeds smoothly without overwhelming the intestine. Key mediators, stimulatory and inhibitory forces. To summarize these regulatory forces: stimulatory, acetylcholine, via the vagus nerve; gastrin, from G cells; and histamine, from enterochromaffin-like cells. Inhibitory, somatostatin, from D cells, suppressing G cells, enterochromaffin-like cells, and parietal cells. Prostaglandins, which also protect the mucosa, and the hormones secretin and cholecystokinin, released by the duodenum in response to acid and fat. Clinical relevance. Why does all of this matter? Conditions of excessive gastric acid, including gastroesophageal reflux disease and peptic ulcer disease, are often managed by medications that block the hydrogen-potassium adenosine triphosphatase or the histamine type 2 receptor. On the opposite end, situations of insufficient acid, such as atrophic gastritis or pernicious anemia, may result in impaired digestion and decreased vitamin B12 absorption. Summary: gastric acid secretion is a beautifully coordinated process. Parietal cells, guided by neural, hormonal, and paracrine signals, release hydrochloric acid in response to food and anticipation of food. The process is driven by stimulatory mediators such as acetylcholine, gastrin, and histamine, and kept in check by somatostatin, prostaglandins, secretin, and cholecystokinin. The balance between these forces ensures the stomach digests food efficiently while protecting its lining from harm. Thank you for joining this episode of our Human Physiology podcast. where today we explored the elegant control of gastric acid in the stomach. Stay tuned for more insights into the marvels of the human body on the Everything Surgery podcast. Dissect the extraordinary.